In this video, I am going to show you the organization of a molecular virology laboratory. I'll show you different kinds of equipments and how they are organized in the laboratory. In the left, you can see this is a chamber of clean bench, and there is two clean bench you can see. And after the clean bench, there is a nitrogen storage bottle. Here we actually use stored nitrogen, liquid nitrogen, and this is the boxes. Actually this flask we use to put nitrogen and to do work, various kinds of work. And here I am going to show you about the PCR machine. There are two PCR machine. This PCR machine, actually here we put our sample in this machine. In this machine only have one chamber, and this machine here have actually two chamber, PCR machine. And from left you can also see here is nano drop machine. This nano drop machine actually used to measure the concentration of DNA, RNA and protein samples. Actually this machine measure the concentration in nanogram level. And also you will find that there is a room, it is called dark room, and this dark room actually used towards the northern blot membrane. Actually this is the boxes we do. Actually it is required to do northern blot and also western blot. And from left, it's left already finished, and from right you can see that there is some fridges and refrigerator. This is the refrigerator of various kinds of temperature, 4 degree, -20 and -80, different kinds of refrigerator you can see. And this is the table, and this is table. Actually this machine for run the gel electrophoresis machine. You can see that here we put sample and do for gel electrophoresis. Here we put such samples, and this is the negative, this is the positive part, and put it here and run the machine. We run the DNA for gel electrophoresis. Here in this fridge, actually normal temperature, put in room temperature various kinds of powders to make different kinds of solution required for various kinds of experiments. And from here in there, okay, this right part already done. Now I can show you the incubator. The incubator actually used to incubate bacterial cells. You can see that the bacterial cells are growing here. Some leaves, actually 37 degrees Celsius incubator to grow E. coli and other bacteria. Here is a centrifuge machine. The centrifuge machine actually centrifuge various kinds of sample, and this machine actually is important machine we use every day. This is the this machine actually used for northern blot. Right now it is closed. This is the circulating machine. It circulates samples like this. Put our sample here for overnight. It requires at least 2 days. And this is the water bath. This is the water bath. Actually similar kinds of incubator. This is the water bath, and this is the pH measuring machine. Here we actually put sample here to measure pH. And this is the metal bath. For high temperature mixing some samples or like this. Okay, this is the dryer. Actually you can see that we can put sample here inside here around 80 degrees Celsius to make dry or various kinds of samples. This is called dryer. And this is the autoclave machine to disinfect our samples and to make media. And this is the tubes, micropipette tubes to be put here before use, disinfection. This is the right part. Another some part I already left, need to show. This is the working place. Here actually students work. This is the another centrifuge machine, and here is some other centrifuge machine. So I show you already left part, and here is the right section. And I also want to show another interesting machine. This machine actually used to grind our leaf samples. Actually this machine is quite interesting. See that this is called a grind machine, and this grind machine actually used to grind our leaves. We collect leaves, and before extracting iron and protein we mix our leaves in this machine. It's very easy to use. And this is the another machine, actually mostly required for it is called orbital shaker. You can see the orbital shaker. Actually during the western boat and some other kinds of activities we use this machine, orbital shaker. And this is the common machine you will find every box, is vortex mixer. Vortex machine, you can see the mixing machine, vortex mixer machine. And I also forget to show another kinds of machine. This is called the shaking bed incubator. Actually it is right now closed because most of the students are on vacation, so they are not using this one. This shaking incubator actually used to shake bacteria. So most of the time we used to shake this bacteria inside 28 or 37 degrees Celsius. And this location in the left we actually used to run RNA gel. This is the similar like protein gel, DNA gel. Here we use this machine like similar, and here is the base kinds of sample like formaldehyde solution and other samples, mobs. And this is the organization where we make gel and run. Okay, this is the nitrogen box, and we have some other equipments, various kinds of equipments here to take picture, and some many other equipments here actually. So with different times we use. And this is the overall organization of our lab. Actually I am working here for last 1 year, and right now I am left this lab as soon as possible, within 1 or 2 months. Okay, this is the very rich, not so rich, it's the medium level laboratory, medium level rich laboratory. Okay, thank you very much guys for watching this video. Okay.